So today we are going to learn about secondary thickening, that is both secondary phloem and secondary xylem. Both can undergo from primary thickening to secondary thickening. How it is being formed, how secondary phloem is formed, how secondary xylem is formed. Last class we had dealt with the in stem how the secondary thickening and secondary phloem secondary phloem and secondary xylem is formed. So this is page number 72. You can see how the thickening are happening, and page number that is 66. From page number 66 to page number 70 we will delve, and some portion of 72 also we will delve in this class. Okay. So what is secondary phloem? Is formed by the tangential division of cambial cell. Oh, hope all of you know where is the location of cambium. Cambium എവിടെയാണ് കാണുന്നത്? What is the function of secondary phloem? It helps in the conduction of food. Okay, basically carbohydrates and proteins. So carbohydrates and proteins are the one which you can see very nicely. Okay, so then this secondary phloem are formed by the fusiform initials. We have dealt two type of initials. You can see fusiform initials and ray initials. And the secondary phloem is formed by fusiform initials. Spindle shape. Hope you remember the spindle shaped fusiform cells. It consists of this different types of secondary phloem are sieve tube, companion cell, phloem parenchyma, and phloem fibers. Okay. And this phloem parenchyma, they are very light, they are very conductive and helps in storage function. What about fibers? Fibers helps in the mechanical strength, give mechanical strength to the plant. Now we know the ray initials. Ray initials helps in the formation of medullary rays. They cross both xylem and phloem. So here phloem rays is formed from ray initials. What is the function? It helps in the conduction of food from phloem to the cambium and then to the xylem. Why? Because these ray initials are running both in the xylem and both in the phloem. Okay. So next we will move with the periderm formation. So before the periderm formation, you can just see this diagram. See how much content the secondary phloem is producing. Only very little, no? And what about the secondary xylem? Xylem is produced in a very large manner, isn't it? You can see lot of xylem content. Okay, the center portion is the vascular cambium. It can be multi-layered or it can be single-layered. See, inside the wood you can see the secondary xylem. This elongated thing is the medullary rays. Medullary rays can pass through the both secondary xylem and secondary phloem. Okay. Now we will move with periderm. Another important portion where you can see the periderm. You can see it on the external surface. Okay, external surface you can see the periderm. Why there is a periderm? Endinengane periderm vandathu? Why? Because there is a pressure inside our stem. It is because of the pressure in the cortical region. Because the xylem cells is dividing continuously. So what happened? The outer wall will get ruptured. So this rupturing of the epidermis is replaced by a protective layer called as cork cambium or Phellogen. Remember that term, Phellogen. Okay, another name of Phellogen is cork cambium. See the diagram? Kando cork cambium kando. Cork cambium by the rupturing of the epidermis. Now another interesting feature is that this Phellogen, no, that is a cork cambium, no, it will divide in a two direction. Okay, it is a live cell, it is a living cell, it is a rectangular cell. Okay, but what is happening over here? This Phellogen will produce some cells outside and some cells inside. See some cells it is been producing outside, and that is called as the cork or the phellem. And some cells are produced where? Inside, right? Some cells are produced inside. That is the secondary cortex or Phelloderm. Okay, secondary cortex or Phelloderm. Like this you can see it. Okay, is that clear? So this is how you can understand the where you can observe your Phelloderm, Phellogen and the Phellem. Okay, remember that this Phellogen is producing cork outside and Phelloderm inside. Okay. So what is how the formation of periderm take place? The formation of periderm take place by the how we have already discussed. This three structure together is called as the periderm, right? So you know the location of the Phellogen, it will be in the center. Top you can see Phellum, that's a top layer, and below you can see the Phelloderm layer. Okay, so in this is a bark, this is a bark, that is the tissue that is seen outside the Phellogen. See, this is the Phellogen. Here you can see the bark region. Okay, bark region. Actually it is same as Phellum. Okay, this cork layer now, that is itself is the bark. See, this is the bark. This is a dead cell. There is no supply of food and water over here. Okay, they are the dead cell. They are seen outside Phellogen. What is a lenticel then? Lenticels are raised pores that is seen on the surface of the stem for the exchange of gases. For the exchange of gases. They are seen beneath the stomata, down the stomata. Stomata താഴെയായിട്ടാണ്. See, they are colorless, loosely arranged cells. See, they are colorless, loosely arranged cells. They are also called as complementary cell. So complementary cell means what? It is the colorless, loosely arranged cell that is seen in the lenticels. They are very thin-walled, and you can see intercellular spaces. See the intercellular spaces, lot of intercellular spaces is there. Here also you can see the Phellogen, Phellum, and Phelloderm. These three together is called as periderm. Remember that point. Okay, these all are seen on the bark, stem bark, right? Now we are moving to the secondary thickening in dicot root. So next we are moving to, stem is over now. Now we are moving to secondary thickening in dicot root. They are done by the vascular cambium and core cambium. They are basically done by whom? Done by vascular cambium, cork cambium. Okay. Root contain radial, hope you know it, radial exarch like a vascular condition, like this, isn't it? Xylem alternating with phloem. You can see like this. This is the radial arrangement, xylem alternating with the phloem. Okay. So this root contain radial like arrangement. This is the cross section of the dicot root. See, this is the big one is called as metaxylem, and the small one is called as what? Protoxylem. Okay, in between you can see the phloem. See, this is a phloem. This is like exarch condition. Exarch means what? Protoxylem towards the periphery and metaxylem towards the center. So this is how the root look like. Now we will move to the cambial activity. There we in stem secondary thickening, we have dealt with the cambial activities formed by the fusion of interfascicular and fascicular cambium. But what about this one, about the root? There is a connective or conjunctive tissue that is seen below the phloem. If this is the phloem, and this is the xylem. This is the phloem and this is the xylem. Here some conjunctive tissue is there. They form what? They form meristematic. Okay, they form meristematic. So cells of, not only that, cells of pericycle. Pericycle is in the below the endodermis, right? If this is the stellar region, this is the endodermis, and just below that you can see the pericycle. This pericycle also becomes what? Pericycle also become meristematic. Okay. So they are seen very close to the protoxylem. So these two meristematic one, this is the first one and this is the second one. These two meristematic together form a wavy band of cambium. They together fuse together to form a wavy band of. Material and the stemmiൽ fascicular and interfascicular cambium will fuse together. Okay, form a cambial ring. Here you can see some of the cells below the phloem and some of the cells below the pericycle form meristematic. Okay, so the cambium, cambium will produce, or they will divide and produce. Outside they will give phloem and inside they will give what? They will give xylem, right? This is the difference. It cuts inside and outside. So more it cuts inside, that is the more it produces, more cells are produced inside, and it helps because of the very pressure inside. It pushes the phloem outward. Okay, then there is lot of pressure. This is the exarch like xylem. Okay, this is the exarch-like xylem. You can see that this radial-like arrangement, protoxylem and metaxylem. Metaxylem in the center and protoxylem towards the periphery. These are the phloem. See the cambial ring formation. This is the cambial. Wavy-like cambial ring is formed by the fusion of meristematic tissues that is present below the phloem and water below the protoxylem. Okay. See how it is being expanded. This is the medullary rays are arising. See how it is being expanding. The xylem is being produced more when compared to the phloem. So when we look into the diagram, you can see only very little amount of phloem is being produced, but the xylem are produced in a very larger amount. Okay, this is the location of a vascular venous. See the rectangular like cells, that is the vascular vein. So this is the primary growth, this is the secondary growth. This is this primary, starting growth. See the XR condition. But when we move to the secondary condition, see how much vessels are formed. See, very big metaxylems you can see. These are the phloem, this is the cortical region, this is the cambial region, and you can see some phloem also. Okay, these all are xylem. Okay, these all are xylem with medullary rays. So now we move to secondary xylem. I have already told you the cambium will cut inside the xylem, means inner surface xylem. Okay, and it can be, they are very active. It produces big cells, large vessels, xylem parenchyma and xylem fiber. See this big one now? That is the metaxylem or the vessels, xylem vessels, because the cambium is very active there. Okay, what about secondary phloem? It is seen outer side. And we can see different types of phloem. That is C2, companion cell, more parenchyma and less fiber. So this is very inactive, no? Not that much active when compared to that of xylem. Okay, now here also there is peridot. But when when peridot is formed, when root will increase its size, no, the epidermis will rupture. Okay, the epidermis will rupture like this, and this some of the cell in the pericycle, no, in the pericycle region you can see some of the cell. That cell will become meristematic and they produces Phellogen or cambium. You know the position of endodermis? Endodermis, then after that endodermis only you can see the pericycle. Okay, and pericycle is facing our protoxylem. Here you can see the xylem. Okay, this is how the, so these cells, no, this pericycle cells will become meristematic. So that is our, here the same condition, the periderm, how the periderm is formed? By the formation of phellum, phellogen and phelloderm. That I will repeat again. എന്താണ് phellum, phellogen and phelloderm? We know that some cells are being cut outside and inside. See, the phellogen is a core cambium which will cut some cells outside and some cells inside. The outside cell is called as cork. They are very water. This cork is very protective layer, dead cells. But what about the Phellogen? They are very live ones. Okay, Phellogen is very live, and Phelloderm is the one that is seen beneath the Phellogen because they are producing that. They are very much good for the periderm formation. Periderm, protective layer is called as periderm. Okay, so when we talk about the difference between these both, what is the difference between? There is a what in stem you can see circular like a what ring formation. Here you can see wavy like, and here you can see fascicular and interfascicular cambium. But here only everything is what meristem like structures, like meristem. You can see phloem, below the phloem and below the pericycle you can see meristem like structure. They form wavy like structure. Okay, and the periderm is seen on the cortical region. Okay, this periderm, another meaning is, another term is extrastellar in origin. Okay, cortical cells. But what about periderm? Periderm is occurring from pericycle that I have told you. That is intrastellar region. Pericycle is seen just closer to protoxylem. Okay. Now here the amount of cork produced will be more. Here the amount of cork produced will be very less. Okay, lenticels are very prominent. Lenticels are not at all prominent. Okay, so these are some of the questions that has to be attempted during your what after looking this video. I'll send one more video also. It will be very helpful for you. You can learn in a very good manner.